Welcome back, everyone. This is the last chapter of the second section of your textbook, so it generally wraps up the information that your textbook provides on the biochemical basis of LTP and learning and memory. I can hear a collective sigh from some of you who may have broad interests in the neurobiology of learning and memory, but perhaps not so much interest in the molecular and biochemical basis of learning. After all, behavioral neuroscience is a broad discipline that uses many approaches to investigate the biological basis of behavior, ranging from the cellular and biochemical basis to the systems levels of analysis that we talked about in the first lecture for Chapter 2. So if you're in that camp, well, rejoice, because after this chapter, we'll move on to more circuit and system levels of analysis. We will occasionally return to talk about some biochemical supports for these circuit and systems modifications, like visiting with our old friend Krebs in some interesting experiments. But we have, for the most part, now learned the fundamental mechanisms that encode molecular and cellular structural changes in potentiated neurons. In fact, Chapter 8 in your textbook consists simply of a summary of the foregoing 7 chapters. It doesn't really go into any new information whatsoever. So I'm going to play along and provide a brief summary in line with the content of Chapter 8. However, I also want to take advantage of the time that we have available, since Chapter 8 is not such a voluminous read, and tell you about another experiment that I think will nicely bridge our review of cellular and molecular mechanisms in LTP to more circuit levels of analysis of the biological basis of learning. I'm going to tell you about a really amazing experiment that links LTP and learning. I think it's actually a very compelling story. In fact, I often joke that trying to find the synapses or cells that underlie the learning of an event or a skill would be like trying to find a needle in a haystack. You got 86 billion neurons in your brain. Where are you going to find that potentiated synapse? However, at the risk of revealing too much about the experiment again before we get into it, I do feel that they put together a really smart experiment to, in a way, well, almost find that elusive needle. But we'll get to that in a few minutes after I do Dr. Rudy's summary of chapters 1 to 7. Oh, and I know before I even, hardly start to record this lecture, that it will be fairly short. But I'm going to be posting it for Friday of the third week of the course, and I am quite certain that everyone will appreciate having a little bit of a break on a Friday. So here goes. In the summary of chapters 1 to 7 that's offered in chapter 8 of your textbook, there's a diagram with a proposal for 4 stages of LTP, which I've copied onto this slide. Now, I've been referring to these 4 stages loosely as induction or short-term LTP, the early phase of late LTP, the later phase of late LTP, and maintenance of LTP. And you'll find similar listings of the stages in many sources that describe the organization of LTP across time. But you will also find sources that describe it more in line with the description in Chapter 8 of your textbook. And I do actually like this taxonomy that was offered up by Dr. Rudy in Chapter 8. So the first stage of LTP is known as generation or induction. And in this stage, within about a minute after the application of tetanic stimulation, NMDA receptors allow in sufficient calcium to activate kinases that phosphorylate mechanisms that accomplish a variety of potentiating functions in the spine. These mechanisms disassemble actin at the head of the spine, insert GLU-A homotetrameric AMPA receptors into the membrane, translocate these newly inserted receptors to the active zone of the postsynaptic membrane, and temporarily trap these new receptors into the membrane, and also activate the UPS system to degrade excess signaling proteins, thereby limiting the overall magnitude of the potentiation of the spine. The second stage is called stabilization. It starts around 15 or 20 minutes roughly after tetanic stimulation if we use standard induction protocols in, let's say, a Schaefer collateral to CA1 pyramidal cell preparation. Now, similar timeframes are generally reported for other preparations as well. But in this 15 or 20 minute time frame, calcium levels continue to rise as calcium-induced calcium release is triggered by the NMDA receptor mediated calcium influx and by the actions of BDNF, overall impacting ryanodine receptors in the endoplasmic reticulum of the spine. And then the metabotropic glutamate receptors, the mGluR1s, they mediate increases in inositol triphosphate signaling that activates calcium release through IP3 receptors in the endoplasmic reticulum of the dendrite and somewhat into the spine. The stage 2 box in this figure from your textbook summary doesn't highlight this, but these actions also initiate local translation of constitutively available mRNA that support the continued provision of signaling molecules like kinases and AMPA receptors. Now, this does appear to be recognized by the vertical brown box that shows which stages involve biosynthetic actions. There's also movement of actin that causes the neck to retract and the spine head to start to enlarge. The third stage is consolidation, which requires from 2 to 4 hours to initiate in a standard preparation. This is the stage that is supported by genomic cascades of signaling. BDNF and mGluR activity continues to hold plasticity, to hold postsynaptic calcium concentrations at an elevated peak. But then signals arrive at the nucleus, and genomic transcription of plasticity products begins to provide specific mRNA and proteins that are targeted in large granules out to the dendrites to be captured and used at the activated spines. The spines that were tagged by the stimulation are capable of capturing relevant proteins, and they have a fully activated cascade of mTOR signaling now to promote translation of top mRNAs. In other words, they are fully engaged in translating the relevant mRNA plasticity products from the genomic signaling. Furthermore, during the consolidation stage, the UPS system is engaged in degrading repressor proteins that would otherwise limit and perhaps slow down the genomic cascades that are responsible for the consolidation of the potentiation of stimulated spines. And of course, the last stage is maintenance over the long term that could range from days to weeks to even years or a lifetime. And in the intact animal, this is mediated by protein kinase M-zeta, or PKM-zeta. Of course, the function of this memory molecule is essential for maintaining the ongoing and persistent presence of phosphorylated GLU-A2 containing heterotetramers in the postsynaptic membrane, by supporting the exocytosis and insertion of these receptors and reducing the endocytosis and removal of these receptors in the face of constant molecular turnover. And lastly, let's address the vertical bars at the sides of the figure, including the brown one that I mentioned a few minutes ago. But I'll start with the blue vertical box that points out that post-translational modifications are involved in all 4 stages of LTP. And of course, then by inference, all 4 are implicated in the neurobiology of learning and memory in the real world. Of course, in stage 1, post-translational processing, mostly kinase-mediated phosphorylation, is pretty much the central event. And there's no evidence of significant transcriptional or translational activities at this stage. And clearly, post-translational processing activities continue on through all of the rest of the stages, even including lifelong maintenance, as we have to keep adjusting to compensate for molecular turnover to replace GluA2 heterotetramers and to keep the potential to respond to input at an elevated level. The vertical brown bar on the left points out that transcription and translation start around the 2nd stage, although I would argue that even if genomic signaling to increase specific transcriptional activities may start in stage 2, it's not extensive enough during this stage to promote any functional consequences just yet. And indeed, translational activity must endure even through the 4th long-term maintenance stage, because protein kinase M-zeta is a self-replicating, constitutively active signal at potentiated spines that promotes its own translation from mRNA to keep the potentiation going. So that covers the summary offered in Chapter 8 of the textbook. Now let's do the bridge. And I'm going to talk to you about a set of very exciting experiments done by Rio Peddotti and collaborators, exploring a link between LTP and circuits that mediate learning of behaviors. I'm going to start the story of this experiment by talking about handedness. We all know that some people are right-handed and some people are left-handed. And of course, this doesn't mean that they cannot use the contralateral limb. It just means that they have a preferred and more skilled hand and arm for engaging in motor tasks. And we also know that the left somatosensory cortex receives input from the right hand and arm, and the left motor cortex controls the right hand and arm, and vice versa, at least for the most part. There is some crossover. Well, these basic biological characteristics are very well documented in humans. So would it surprise you to know that the same is kind of true for rats? Each rat seems to have a preferred forelimb that it uses for reaching and grasping, and it's controlled by the contralateral hemisphere. Now, I don't think their degree of lateralization is quite as extensive as ours, and they don't prefer one forelimb as much as most of us do, probably mostly because they don't have the specialized skilled control of the hand, or in this case the paw, like we do. However, if you place a rat into an experimental task situation where the animal has to reach and grasp with a forepaw, they will reliably use one preferred forepaw over the other. So that's what Rio Peddotti and collaborators did. They placed hungry rats into an acrylic arena with a narrow slot at one end, shown right here. They then they then placed a nugget of food at the limit of the rat's reach so that it had to extend its paw through the narrow slot, pick up the tasty morsel, and bring it back through the narrow slot to eat it without dropping it. Now, for a rat, that's an extremely challenging task, testing the limit of its potential to learn a fine motor skill. But note that it is a skill a rat can learn. My dog, for example, is a very intelligent creature by comparison, but he can't manage to pick up any small objects with his forepaws. He can merely bat things about as he doesn't have dexterous little hands. But rats are actually quite capable of manipulating objects with their forepaws, as you can see in the picture. They're indeed actually quite dexterous. So, Riupodati trained the rats for 5 days and found that the rats' abilities reached asymptotic performance after about 3 days. The dependent measure in this experiment was a ratio of the amount of food consumed over the total amount of food available. So the food pellets were carefully weighed before the experiment. The rat might successfully consume some of it and drop some of it where it would fall down into a tray below the arena. So the experimenters simply subtract the spillage from the amount of food offered, and they have a measure of the success of the animal at consuming the pasta. Okay, I'm going to take a brief time out to tell you about something that I find objectionable in this experiment. If you look at the learning curve on the graph, they report how many rats were tested on each day of the experiment. Now that's good, but what you can see here is there's a sudden drop from 95 animals on days 1 and 2 to 57 animals on day 3 and a few less still on days 4 and 5. Now, the authors of this research report that was published in the journal Science, they don't explain why the numbers dropped, but suddenly the curve flattened on days 3 to 5. Did they perhaps eliminate all animals that were unable to perform the task? I don't actually think that would be an unreasonable thing to do, as any rats that can't learn would provide meaningless distortions in the data. Or perhaps did they drop all the animals from the experiment that didn't actually use a preferred forepaw and rather switched back and forth between paws? That would also distort the outcome of the experiment, which I think you'll appreciate better after I show you a little bit more of the methodology. So, I don't think that what they did was particularly unreasonable, as they kept the rats that had learned the task and did it at a high level of performance, up to about 90% efficiency,